Welcome to the PubMed Daily Review for March 12th. Today we scanned 6,557 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, Plasma Phosphorylated Tau 217 and Incident Mild Cognitive Impairment and Dementia in Older Women by Aladdin H. Shadiab and Linda K. McAvoy from Herbert Wertheim School of Public Health and Human Longevity Science, University of California San Diego, La Jolla. Let's break down its key contributions. We know that Alzheimer's disease is a growing concern, and finding reliable ways to predict cognitive decline is crucial. While brain imaging and spinal fluid tests exist, blood tests offer a more accessible option. One promising marker is plasma phosphorylated Tau 217, or p-Tau 217. This study aimed to see if p-Tau 217 could predict the development of mild cognitive impairment or dementia in older women and explore if factors like age, race, genetics, or hormone therapy use influenced this prediction. To answer this, researchers analyzed data from over 2,700 women enrolled in the Women's Health Initiative memory study. These women were cognitively healthy at the start and were followed for up to 25 years. Their baseline blood samples were tested for p-Tau 217 levels. The main goal was to see if higher p-Tau 217 levels predicted the later development of mild cognitive impairment or dementia. The results were striking. Each 1 standard deviation increase in p-Tau 217 was linked to a 2.43 times higher risk of developing MCI or dementia. This association was even stronger for dementia alone. Importantly, the study found that these associations were not uniform across all participants. P-Tau 217 was a stronger predictor in white women compared to black women, in APOE4 carriers, and in women over 70 years old. Hormone therapy, specifically estrogen plus progestin, also appeared to modify the association with dementia risk. So, what does this mean for us? This study highlights plasma p-Tau 217 as a powerful, accessible biomarker for predicting cognitive decline. However, it also emphasizes that we can't look at this marker in isolation. Factors like a person's race, age, genetic background, and even hormone therapy use can affect how strongly p-Tau 217 predicts risk. This suggests that future approaches to risk assessment and potential interventions will need to be personalized, taking these important individual differences into account. Up next, our second paper out of 5, Plasma Proteomic Dynamics Preceding Glaucoma reveal a 15-year pre-diagnostic window. Causal insights and predictive utility in 45,850 participants by Inka Zhao and Jihong Wu from Department of Ophthalmology, I&ENT Hospital, College of Medicine, Fudan University, Shanghai, China. Let's break down its key contributions. Glaucoma is a silent thief of sight, often progressing unnoticed until significant vision loss occurs. Early detection is key to preserving vision, but current methods fall short. This study aimed to uncover subtle, early warning signs in the blood, specifically looking at proteins, to see if we could predict glaucoma years in advance and understand what might be causing it. Researchers analyzed plasma samples from over 45,000 individuals in the UK Biobank, none of whom had glaucoma at the start. They measured the levels of nearly 3,000 proteins in their blood. Over a median follow-up of more than 16 years, they tracked who developed glaucoma. They then used statistical models to see which proteins were linked to glaucoma risk and if these changes appeared years before diagnosis. The study found 136 proteins significantly associated with glaucoma risk, with EDA2 are showing a strong link. Remarkably, these proteomic changes began appearing 12 to 15 years before a clinical diagnosis. Furthermore, machine learning models using these protein signatures achieved an impressive area under the curve of 0.803 for predicting glaucoma up to 2 years in advance, a substantial improvement over models based on demographics alone. This research opens a new frontier in glaucoma detection, revealing a surprisingly long window where the disease's molecular footprint is present in the blood. The identification of causal proteins like LTBP2 not only deepens our understanding of how glaucoma develops but also points towards potential new ways to treat it. Ultimately, these findings suggest that proteomic analysis could revolutionize glaucoma screening, allowing for earlier interventions and better patient outcomes. Up next, our third paper out of 5, Single-Cell Resolution of an Open Chromatin Signature in Persister Tumor Cells by Mihai Gabriel Dumbrava and Alexander Gaspermaya from Division of Experimental Pathology, Department of Lab Medicine and Pathology, Mayo Clinic, Rochester, Minnesota, USA. Let's break down its key contributions. Cancer recurrence is a major challenge in oncology, often stemming from a small group of tumor cells that survive treatment. These persister cells can then regrow the tumor, leading to relapse. Identifying the unique characteristics of these persister cells is key to finding new ways to eliminate them and prevent cancer from coming back. To uncover the secrets of persister cells, the researchers employed advanced single-nucleus multiomic profiling. They analyzed samples from ovarian cancer patients, comparing normal tissue, treatment-naive tumors, and tumors after chemotherapy. This allowed them to pinpoint specific molecular features, particularly in the chromatin landscape, that characterize these resilient cells. The study successfully identified a distinct open chromatin signature present in persister tumor cells. This signature wasn't just descriptive, it proved to be a strong predictor of how well patients would respond to chemotherapy. Furthermore, these persister cells showed signs of being primed for expression of certain genes even before treatment, along with altered cell cycle and stress response pathways. This research reveals that tumor cells possess an intrinsic program that makes them tolerant to chemotherapy, characterized by a specific chromatin signature. This signature is detectable early on and can predict treatment outcomes. Importantly, these findings open up new avenues for therapy, suggesting that targeting chromatin-modifying agents could be a promising strategy to prevent cancer recurrence. Up next, our fourth paper out of 5, Phosphorylated tau exhibits antimicrobial activity capable of neutralizing herpes simplex virus 1 infectivity in human neurons by William A. Eimer and Rudolf E. Tanzi from Aging Research Unit, Henry and Allison McCants Center for Brain Health, Mass General Institute for Neurodegenerative Disease, Charlestown, Massachusetts, USA. Let's break down its key contributions. We often hear about tau protein in the context of devastating neurodegenerative diseases like Alzheimer's, where it misfolds and aggregates. But what if these pathological changes are actually part of a protective mechanism? This research explores the surprising idea that hyperphosphorylated tau might be an ancient defense system in our brain, fighting off infections. To test this, the researchers used human neuronal cell cultures and the herpes simplex virus type 1, or HSV1. They treated the neurons with phosphorylated tau, then infected them with the virus. Using advanced techniques like confocal microscopy and electron microscopy, they observed how tau interacted with the virus at a molecular level, looking for evidence of viral neutralization and binding. The results were quite striking. Pre-treatment with phosphorylated tau significantly reduced the number of HSV1 plaques and single-cell infections in the neurons. Furthermore, p-tau showed a much stronger binding affinity to the HSV1 viral capsid compared to unphosphorylated tau. They also observed increased p-tau aggregation within infected neurons, reinforcing its role in the host response. This study suggests we need to rethink our understanding of tau. Instead of just a marker of disease, hyperphosphorylated tau might be a crucial part of our brain's defense against infections like HSV1. This opens up exciting new avenues for research, potentially leading to novel therapeutic approaches for neurodegenerative diseases by harnessing the brain's own immune defenses. Up next, our fifth paper out of 5, Inhibition of macrophage pyroptosis protects against sepsis-induced injury to the alveolar epithelial barrier by Min Wang and Dan Zhang from Department of Nephrology, Changdong Hospital affiliated with Xiamen University, Xiamen, China. Let's break down its key contributions. Acute lung injury, or ALI, is a serious condition where the tiny air sacs in our lungs, the alveoli, become damaged. This damage often involves the barrier formed by cells lining these air sacs, and it's largely driven by excessive inflammation. While we have supportive care, we lack treatments that specifically target the root causes. We know that a process called pyroptosis in macrophages, a type of immune cell, can contribute to lung injury, but how exactly it affects the alveolar barrier in sepsis wasn't fully understood. To investigate this, the researchers used both cell cultures and a mouse model. In cell cultures, they stimulated macrophages with LPS, a component of bacteria, and then treated some with MCC950, a drug that blocks the NLRP3 inflammasome, a key player in pyroptosis. They then exposed lung epithelial cells to the conditioned medium from these macrophages to see the effect on the barrier. In mice, they induced sepsis-like lung injury with LPS and treated some with MCC950. They measured various markers of inflammation, cell death, and barrier integrity, as well as overall lung damage. The results were quite striking. LPS stimulation robustly activated pyroptosis in macrophages, as shown by increased caspase-1 activity and inflammatory cytokine release. This activation led to significant damage to the alveolar epithelial barrier in cell cultures. However, when macrophages were pretreated with MCC950 to inhibit pyroptosis, this damage was significantly reduced. Crucially, in the mouse model, MCC950 treatment markedly alleviated lung injury, reducing edema and barrier leakage. These findings strongly suggest that blocking macrophage pyroptosis is protective. So, what does this mean for us? This study clearly identifies macrophage pyroptosis as a critical driver of lung barrier damage in sepsis-induced ALI. Importantly, it highlights that targeting this specific pathway, rather than broad inflammation, could be a more effective and precise therapeutic strategy. While these findings are promising, further studies are needed to confirm these results in more complex models before we can consider this for human patients. Nevertheless, this opens an exciting new avenue for treating a devastating condition.